Good morning, good afternoon, everyone. Thanks for joining us today. Today, we're going to be talking around the 2025 Dotmatics Innovation Showcase. Today, we're really excited to explore how we're connecting science, data, and decision-making to shape a better future together. Next slide. Today's presentation may contain some forward-looking statements, including projections, estimates, and other information that reflect our current expectations, more of our standard harbor statement there. Next slide, please. And I'm Tamara Adams. I'm Chief Revenue Officer here at Dotmatics, and it's my pleasure today to introduce today's speakers. Thomas Swala, Chief Executive Officer for Dotmatics, Kaleem Saliba, Chief Product Officer, and Phil Motney, VP President of Science and Technology. During today's session, we'll be answering your questions live through the chat window, and I'll be monitoring it behind the scenes, so feel free to submit them at any time. And if for some reason we don't get to everything today in the chat window, we'll be sure to follow up with you directly after the webinar. So with that, I'm going to turn it over to Thomas to get us started.
Yeah, I appreciate it, Tammy. And thank you guys all for joining, whether you're watching it live in real time or whether you're going to be rewatching this afterwards. And I think it's just really important for us. And part of the reason why we do this is not only to show kind of how we are trying to push science forward, but it's to hold ourselves accountable too. And those of you that have heard me speak over the past 3 years, 2 years, 18 months, you will kind of know the strategy that we've employed here at Dotmatics. And you're going to see the progress against it. And the strategy hasn't changed while we continue to add capabilities and products and talent to our organization. We continue to maintain that same strategy. So hopefully, like I said, this is holding us accountable. You hold us accountable, and you'll see the progress that we're making kind of every 3, 6, and 9 months. So I'm happy to be here today. So I just want to start with this first slide, and I think we've all seen this, right? This is Eroom's law. And I think the challenging thing when I look at this slide and I see the word Eroom's law is every other industry, aerospace, semiconductor, electronic, mobility, financial services, Moore's law is what drives their ecosystem. Moore's law. Yet here we're still stuck in Eroom's law. And I know this is the business problem that many of us are trying to solve is how can we be a part of turning this around? How do we be a part of driving efficiency? How do we be a part of discovering things quicker? And how do we move things through the R&D funnel faster? And I think there's a belief system, I certainly have it, that software, AI, accelerated compute, simulation, collaboration, and all the things that you guys know are going to help turn Eroom's law around. So if you go to the next slide and you think about some of the reasons why this is, and you guys live it every day. I hear it from customers every day, but the landscape is extremely fragmented. If we look at the software that exists in the R&D section, it is much more fragmented than any other industry. And I think it's one of the reasons why I think our industry has been about a decade behind other industries kind of going through this digital transformation is it's incredibly fragmented, which leads to siloed data. There's also these complex architecture needs that exist within your organizations. You have systems and workflows and processes that have been around for a long time. But by the way, all other industries have gone through something like this. I will say, and I think most people will acknowledge that our ecosystem, given biology, is harder. The fragmentation is harder. But there's this, the saying I go to, which water will find a crevice. So we know the direction of travel, and it's how are we going to get there? And you're going to see today how Dotmatics is kind of pushing the ecosystem forward. So I won't spend a lot of time here, but I want everybody to realize the breadth of capabilities, solutions, and products that exist within Dotmatics. And many of you use them today, whether it's Prism, Geneious, or Versidian, or Omic and flow cytometry. And that's against the backdrop of an informatics platform. And we have platforms that are ELNs specifically for mid and large tier enterprises. We have informatics platforms that are more suited for university and academic settings as well. But I think what we're going to focus on a lot today is the really core innovation that we decided about 24 months ago to really make a big bet on, and that's Luma. And you've probably heard about Luma before. You might have seen demos about it, but you're going to see more today how Luma, when you combine it with all of the solutions we have, can really push science forward. And this is exactly what I was talking about. I think if you look at where we are in the digital transformation journey, you have the left side of the screen where there's this individual fragmentations, or people refer to them as point solutions, that are really good at doing a specific job. But we know all of these tools exist within a workflow of some modality trying to actually make some discovery. And so where we are today is right in the middle. We're trying to integrate all of these tools today. And I think if you think about one of the biggest challenges is integration and interoperability in our ecosystem, where there just hasn't been enough traction around standards and agreements of data models and APIs. But the reality is that is what's needed. You need integration and interoperability to get to the right-hand side of the screen, which is how do you actually collaborate and automate and leverage AI for drug discovery? And Dotmatics' answer to that is when we take Luma and combine it with all of the best-in-class software applications to be had. It's what will really drive efficiency into the future. And as I talked about, and I think it's important to show this slide, I think folks that haven't lived in manufacturing or in automotive or in semiconductors sometimes are surprised by that. But what this slide shows is from 2019 to today and where it's projected to go in 2029, it's the software spend. The software spend relative to other very research intensive industries. And you can see that the biopharma world is lagging behind all of the other industries in terms of software. And we know, even in our ecosystem, digital transformation, AI, accelerating compute, all of them are dependent upon software. So we're a little bit further behind, but we know that software spend will increase because it's increased everywhere. And ultimately, software is deflationary and it will drive efficiency. And yes, there are a lot of other things, organizational workflows, change management, that come into play. But one thing is true is as industries move through their transformation process, software becomes a bigger percentage of spend over time. And the last thing I wanted to note, and I wanted to address it, and I think a lot of people have probably seen it, is the proposed transaction between Siemens and Dotmatics. And the reason that I say proposed is I think it's important to realize that as of today, we are still owned by our current shareholders, and we're in this period of regulatory approval. So there's a handful of countries and regulators that need to approve this transaction before we actually get started on day one. But I think it's really important to note that what we're doing in R&D, taking basically all of these applications and tying them together with a digital thread. The thesis of Siemens, and this is directly out of their investor relations deck, is how do we take that digital thread and tie it not only from R&D, but into manufacturing as well. And I think everybody knows the challenges that happens with different platforms and software and technology between basic research and manufacturing, and all of the steps it needs to take. And I think if you take a look at Siemens, that, by the way, participates in those other industries, automotive, semiconductor. If you look at the pattern that those other industries have gone through, it is a very similar pattern. So we will be a part of their software division, which is a very large software company that leverages their simulation and AI capabilities that can extend from research all the way through to manufacturing. So solving both problems around how do we do multimodal research in an AI first world, and then how do we move that molecule through to manufacturing is the thesis. And again, day zero hasn't even started on the acquisition. And I think it's important for folks to note that, but hopefully over the next several months that will take place. But I think it's important to note the thesis and the leverage and the capabilities that we get from combining Dotmatics with Siemens expertise as well. So with that, I'm going to throw it over to Kaleem to continue to kind of walk us through Luma.
Thanks, Thomas, for that great insight into the industry and the types of challenges that our customers are facing. I'm really excited to be able to talk to you all about Dotmatics Luma today, the scientific intelligence platform that's at the heart of solving a lot of these challenges for our customers and the industry. Dotmatics Luma powers the design, make, test, decide lifecycle that is core to scientific R&D. And it does that by really bringing four key areas together. The first are scientific and IT disciplines across a wide range of personas. The second are scientific techniques, leveraging the Dotmatics industry-leading scientific applications, but integrated with Luma via repeatable patterns that allows our customers to integrate third-party software as well. The third are R&D modalities. As a multimodal platform, Luma allows our customers to leverage one or multiple modalities in addressing a particular need, which is really important to the kind of science that happens today. And then this inevitably takes our customers across a wide range of data types and formats. And as a robust and comprehensive data platform, Luma can handle all of these seamlessly and gracefully. And there are four key benefits that I want to make sure you all walk away with today. And these benefits together underpin the foundation of the unified platform we are creating for process modeling, data automation, and discovery. The first benefit is flexible precision in representation. Whether you're talking about biologics or small molecules or conjugates or other molecule types, Luma can represent these things with the exacting level of precision that's required for good science. And it does this via flexible yet governed data models. The second key benefit is process and data alignment for AI. Luma serializes dry and wet lab tasks in real time, morphing to the twists required in research, but also supporting the more rigorous and prescriptive methods that are required for development. And it does this while aligning data precisely at every step, increasing the accuracy of AI and increasing the efficiency with which it can be deployed. The third is our specialized scientific capabilities. By embedding this industry-leading software within our portfolio, within orchestrated Luma workflows, our customers can get both the breadth and the depth of scientific excellence that's required to succeed today. And the fourth is multimodal self-service agility. The data sets, the requirements, the business needs, they are not standing still, and neither should your software. By leveraging key patterns, open APIs, event-based integration, and low-code configuration, the software can evolve at the pace that your requirements and business demands. And Luma ultimately does this by bringing together a number of foundational capabilities that are purpose-built for science. This is a high-level overview that Phil will go into more detail on. I just want to touch upon the basics. At the very core, you have a low-code application layer. This allows our customers to create and configure their own packages of functionality that do exactly what they want using a low-code interface. And it's built on scientifically aware storage. So we've partnered with Databricks, the world's leading data lakehouse, and invested substantial IP on top of Databricks to build out and scientifically enable it within this low-code ecosystem of broader configuration. On the outer ring, you can see the different configuration categories that our customers are empowered to leverage within their apps. Instrument and data integration allows them to get at any data set, wherever it might exist. Data management and processing takes semi and unstructured data, flows it through data flows, and then maps it to harmonized and well-connected models that can be reasoned about. Models that connect traditionally disparate and siloed data sets. Material and ontology management brings an ontology-first approach along with asynchronous multimodal registration to allow our customers to focus on the work that matters while being unimpeded in the efficiency that they're trying to execute with. Adaptive workflows allows our researchers to really embed their creativity within an adaptive structural framework for workflows. And in the development space, lock those down into a more prescriptive and linear representation of what needs to happen. Now, by having all of this data and all of these process steps within Luma applications, the decision support that is made available to our customers is substantial. We provide a low-code drag and drop interface to create your own business intelligence dashboards, charts, reports, along with scientific componentry that we add to the platform based on our broad and deep portfolio of scientific capability. And because data is the hardest part of AI, by having all of this data well connected and at your fingertips, you can seamlessly integrate with your external models and eventually leverage Luma based on the underlying Databricks capabilities to configure, train, and execute models directly within the Luma platform itself. And Luma is fair data compliant, which means you can get at your data whenever and however you want. We have numerous open APIs and governance and security that you can control completely. Another really key component of Luma is Luma Lab Connect, which brings our industry-leading products together with very highly scalable data. Luma Lab Connect is our file-based ingestion mechanism. It has support for hundreds of instruments and growing, and it also supports arbitrary file types, including the files that are generated from our and third-party scientific software, things like Prism files. And all of this ultimately, creates the opportunity to do something very powerful. And Thomas mentioned it. And this is kind of it at the next level of detail, where Luma can automatically create these digital threads, which centralize and harmonize process and data in real time as you work. And that word real time is very important, because the norm today is retrospective analysis and data collation by trying to pull together Excel spreadsheets and database exports and data warehouse extracts and trying to make sense of it all. And by the time that's been done, the data is often stale, errors have been made, and if you're even able to do it, it's a very laborious process to try and make sense of it. Luma shifts the paradigm from retrospective to proactive real-time decision-making. And it does this by creating these threads. What you see in the center here is a very high-level representation of a biologics workflow. It applies equally to other workflow types. And you see a multitude of different personas, stakeholders, really different functional departments engaged with the software. Each person has the benefit of the upstream context that was created for them and produces outputs that represent the downstream context for the scientists that will work on what comes next. This creates digital threads of data and digital threads of process, because each of these actors is creating and consuming data and also working through process steps. And Luma automatically tracks and harmonizes the lineage of process and data in a way that is completely connected to each other, so you can reason about it in real time. This leads to better decisions, and it's also a really important precursor to lab in a loop. Lab in a loop, done in a retrospective way, isn't a very contiguous loop. By having a real-time digital thread, you can more seamlessly feed the outputs of this process back into the inputs and refine the machine learning models that are involved in the overall end-to-end workflow. Now, I'm going to spend a little bit of time diving into some of the more specific challenges that some of these personas face before handing it over to my colleague Phil, who will show you some of this stuff in action. Protein engineers have no higher order representations of proteins today and no linkage to their related data sets. This makes protein engineering very complex, very time consuming, and frankly, very siloed. The benefit with Luma is we are bringing together to the table in our partnership with BioGlyph a standardized higher order representation of proteins and links to related data sets that automatically drive greater efficiency and context. Now, what you see pictured on the right here is such a representation. This is a multi-specific antibody represented by a collection of building blocks, domains, and formats. And this automatically gets registered and linked to all downstream activity that is related to it, critical to being the tip, the start of that digital thread. Cloning expression purification groups have a multitude of different materials and entity types that they have to track across a fairly sophisticated life cycle. Today, this is often done manually, tracked manually and registered manually, which creates significant logistical challenges. With Luma, the benefit by leveraging the adaptive workflow capability is automatic linking and tracking of these entities, which increases the reliability and traceability of the overall process. This is particularly exciting because you can see the digital thread in a quite literal manifestation taking place in the bottom right of this screenshot, where you see the different materials linked to each other. And these linkages are created automatically as scientists work. They do not have to be created retroactively. And in the top screenshot, you can see a view of our adaptive workflows, which have inputs, unit operations that can be executed against those inputs, and then outputs. This is what accepts the upstream context from other scientists and produces the downstream context needed to continue that digital thread of work. For analytical scientists, the challenge, again, you'll notice a pattern here, where the challenges are often manual and the benefits are brought about by automating a lot of that which is manual. Manual annotation of LC-MS data and manual collation of silo characterization data leads to lost time. The benefit with Luma is an automatic annotation of that data and real-time collation of the related data sets, leading to better decisions. And you can see two example screenshots of this happening on the right here. Again, this is brought together in partnership with BioGlyph. Assay scientists have a particularly important challenge because it's not just about manual pre-processing of the data in Excel. There's also oftentimes copy and pasting, not just copy and pasting, but a transposition of data formats during the copy and pasting process into external tools. This creates a significant challenge, lots of room for error, and also requires manual collation of assay results with other relevant data sets so that you can reason about them together. The benefit with Luma is that all of this becomes automated pre-processing of the assay data, bidirectional interoperability with Prism, and automatic association with the related data sets drives greater reproducibility and time savings. And what you see happening on the right here is an assay-focused task. That task has an expression engine that we have built into it. In this case, a percent inhibition calculation has been set up. That's run on the data before the export to Prism, where the graphs are generated. And then really importantly, automatically re-imported into Luma and linked with the task that generated the Prism file and the materials that that Prism file was actually running the assay for. And last but certainly not least, we have our IT and informatics scientist stakeholders. The challenge here is enormous, right? This is where typically the solution has been to do a lot of retrospective analysis and stitching together of lots of disparate data sets. But with the real-time digital threads Luma is bringing together, the benefit is that that large confluence and volume of data is connected and contextualized in real time as the scientists work, which drives timely and informed decisions. I won't go into the detail of these dashboards. My colleague Phil will cover them, but I just want to note about these is that these are connected to the real-time data sets that those digital threads within Luma are producing. So you don't have to do retrospective analysis to set these up. Now, before I hand it over to Phil, I wanted to take a brief moment to give you a view into the composable product portfolio vision that we are working hard to bring to market to power the next generation of multimodal research. And it's composable across two important dimensions. The first dimension are the functional areas, the departments, the people, the oftentimes silos that have to work together to drive the design, make, test, decide cycle forward. The second dimension are modalities. Whether you're working within one modality or trying to combine a multitude of modalities, composability within and across them is really important to being able to have choice and flexibility needed to do the job right. And with the products we're bringing together in the Dotmatics portfolio, we are really giving our customers choice in how they compose their solutions. So whether you have one very specific need within a functional area, or whether you're looking for a comprehensive end-to-end solution for an antibody discovery or protein therapeutic use case, Luma has you covered. And what's great about this is that Luma grows with you. You should see it as an ecosystem where you can start as small or as large as you need to and have the system grow with the choices that you make and the needs that you have as the requirements and as your businesses change. And with that, I'm going to hand it over to Phil. I don't want you to take my word for it. I want you to form your own opinion. We believe this is the most advanced scientific intelligence platform ever delivered, and Phil is going to give you a small taste of it next in his demo. Phil, over to you.
Thank you. Ryan, it looks like I need, there we go, I got it. Let me know when you see my screen.
Got it.
I'm just going into slideshow mode. Okay, hi everybody. So I'm Phil and I'm here to give you the demo portion here. And I'm starting off with my favorite view of Luma. So I like to think of this hub and spoke model. The apps around the outside are all Dotmatics apps. We have Lab Connect for instruments. We have our various scientific apps, FCS Express, Omic, Prism, our notebook. The same is true for third-party apps though. And if you think about this, our ecosystem looks a lot like a customer ecosystem now. And before Luma, we were struggling, as we know you guys are struggling, to make sense of all of this data and really unlock all of this data. So the most important thing that Luma does, in my opinion, is allows all of these apps to push data into Luma. Luma can then put them into a data model that makes sense and is easy to understand. And then all of these apps can pull data back out, but not only their own data, data from the entire ecosystem. So it's this great data flow that allows us to create the great workflows that I'll show you next. So let's have a look at Luma, the app in the middle there. I'll show you some of the architecture and kind of how it all fits together, and then we'll come back and actually do the workflow. So this is Luma, and I want to draw your attention to some of the configuration that's possible in here. In the bottom right, I'm actually in a dashboard, a decision-making dashboard. This is where we want to get you guys to quickly. This is where you can go and drive your projects forward. But let's have a look at the different configuration categories that we've got at the top. I've started in data, again, in my opinion, the most important category that we have. From data, we can do things like create tables and views and relationships between them. We can build our data model. We can choose, where does this data come from? Is it a database? Is it an app? Is it an instrument? Is it a third party? We can take the data in its raw form and we can manipulate and move and massage it and push it up into our data model. As Colleen mentioned, this is an open ecosystem. You can get data out via database queries over JDBC or via API calls. And you can design the experiences that your users will interact with the data in the bottom right here. Users can obviously design their own as well, but you can give them a foundation. Now, to get here, we have to work in the lab. Adaptive workflows allows us to produce digital twins of laboratory processes. I'll focus on the wet lab today, but this is also going to be for the dry lab. You work in a laboratory, of course, you have to register the entities, both the concepts and the batches. This registration system is the first, I think, truly multimodal registration system. So we'll handle small molecules, large molecules, as you'll see today, and all of the wonderful conjugates in between. I'm going to be talking today specifically about antibodies, so I'll show you our protein design module. And then regardless of the type of research that you do, you want to be able to pull in instrument data. Really, any system that produces a file can flow its data into here via Lab Connect. Now, all of this context, all of these pieces I've just talked about are all contained within an app. I'm in my antibody campaign app right now. And as you can see from my app switcher, I can have multiple apps in Luma. An important thing to understand is all this configuration can be wholesale exported and imported. And that's going to make your lives really easy when you guys have to move from dev to test to prod. It's going to make it easier for Dotmatics to make improvements and share those with you. It's just a really modern, well-engineered software platform. I'll come back to the really important relationship between apps a little bit later, but let's go and have a look at the workflow that I'm going to be working on today. Let me just get this out of the way. I'm just going to move to my next app, so I'm ready. Okay, so the workflow that we're going to tackle today is an antibody discovery workflow, a multi-specific antibody discovery workflow. The chevrons on screen here, I know that there's a lot more chevrons, and I know that these chevrons hide a lot of detail underneath them. I also know that you probably don't want to sit here and watch me do a demo of each of these chevrons, so I'm going to break it down a little more simply, and I'll use design, make, test, decide to frame up this demo. And design, make, test, decide is critically important to any kind of research. So you could swap out this for small molecules or for formulations, but we'll focus on multi-specific antibodies today. This portion of the market is generally underserved compared to small molecules, for example. So let's move to the first step, design. So this is our collaboration with BioGlyph. And this is, I think, going to change the industry because, as Kaleem said, it gives us a higher order, a common language to talk about these types of drugs that we're trying to make here. Now, this is a multi-specific project. So part of the project setup is to design or to indicate the targets that I'm trying to operate against here. And these could come from other campaigns. They could come from a monoclonal campaign that's part of this. And we assign these targets different colors, and we use these a bit like Lego blocks to then build up the formats that we want to create in our project. So I already have some registered, but I can use my beautiful design pad here to go and just create a new format at will. This is all done with drag and drop. It all snaps into place. You can see on the right-hand side, sorry, on the left-hand side here, some of the different building block types that we support. And we'll continue to add to these so that we can help you keep pace with the science. Now I can then go and change the specificity, the different targets to match up with the project level targets that I'd set before. So we don't yet have a full antibody. We have the format, and we need to layer over the chains on this format to produce a digital twin of the target antibody that we want to make. But one of the big challenges with this kind of research is that these molecules are actually quite hard to make. Over 50% of these will fail in the lab, and they'll fail for liabilities like coagulation or poor expression, etc. And you may want to engineer around some of those liabilities by creating mutant or child sequences of some of the sequences in your library that perhaps contain too many liabilities. We have a tool to allow you to do that. So if I go back to my dashboard here and I go to my variant analyzer, I've skipped over a few steps here where I've perhaps gone out to Geneious and done my mutations, or you pick your favorite tool to do that. And I've imported the parent sequences, the original ones, and the mutated sequences into my analyzer. And now I can go and view the details of this analysis. So in this analysis, I have 4 or 5 different parent sequences, and when I uploaded all of this, it's done several things. It showed me where the mutations are, and I can choose various numbering schemes to denote that. I can then filter down and look at the parent at the top and all of the mutated sequences underneath. I can drill down and I can go and look at the details of each of these sequences, including the residue detail and including the annotations that I've added as I've uploaded these. And then it's done some really cool work, actually. Now I can do an alignment against the parent here, and I can highlight the mutated residues here in my little map. I can look at a tree map of all of this, and I can always interact with my parents here. But I think the most interesting part here is then we've done some dimensionality reduction. The larger circles here are actually the parents, and the smaller circles are the children. So this allows me to look at the sequence space and the sequence variation that I've generated as part of this analysis. So maybe now some of these mutants have actually worked around some of the liabilities, and I'm ready to go and actually build up the full picture of my antibodies. I'm ready to go and layer over the sequences over my formats. And this is more than just an enumeration problem, because you have to add in the linkers and the hinges, etc. We see people struggling to do this in Excel, partly because of the volume and partly because of that biological smarts that's needed. We've actually solved that with what we call panel design. So in my panel designer, I can take these formats and I can click on them, and then I can start to look at my libraries of chains. And as I click on the various options, it's producing what we call a subpanel underneath. And as I click and add more, it's enumerating all of the possibilities over the choices I've made here. But you can imagine, as you do this across sequences in a project and over vast libraries of sequences, you can soon generate panels that are very large. This is quite a small panel, but once it loads up, I'll show you how we now have a digital twin, a fully specified antibody ready to go. So if I go down into the details here, click on one of my designs, and now can see a fully annotated antibody with all of the different regions, all of the different specificities, all the way down to the residue level. And this is going to really change the game in terms of how we talk about these, how we make sure we're not repeating work, and how we can now organize and layer the data underneath in a really meaningful way, much in the same way that chemistry's had for a long time, being able to draw their structures ahead of synthesis. Okay, let's move on to the wet lab portion of this, and I'm going to divide this up roughly. Again, this is high level, into 2 pieces. The first piece is I'm going to be making constructs that give me the specific chains that I need for my antibody. And then when I move on to my next step, I'm going to co-express these in an expressing cell line to give me my crude antibody. But it turns out before I can do that first step, before I can make these constructs, I actually have to go through another round of design. I have to do some molecular biology to design these constructs. And I'm going to do that in my Geneious platform. We, of course, have Snapgene, or you could use your own tool here. Now we're back to my favorite circle view here. Remember, Luma provides great data flow, which allows great workflow. So in many of the Dotmatics tools, and you'll see this repeated throughout the demo, I can log into Luma. Here I am signed into my Luma tenant. And that allows me to do two things. It allows me to query the panels that I've just designed and pull out the fully annotated sequences or fully annotated regions of sequences. I can then go and combine that with other sequences that I've got, and I can do in silico cloning in here. And I've actually already done this. So I now have a table at the top here with my various constructs. I like to think of the constructs as recipes, and the various regions inside the constructs, I like to think of them as ingredients. Some of them are synthesized in silico, like the inserts, for example. Now, Colleen mentioned fair data before, and we found that some of our customers are struggling at this point. They're exporting this data into Excel, or they're putting these files in a shared drive. They're trying to use PowerPoint and email to communicate with the wet lab and say, hey, I'm ready to action this. I'm ready to make these. So we've broken down that data silo by, again, via the login to Luma. I can just choose the constructs that I'd like to send across. And then with a click of a button, send these designs across to my wet lab to make. So this is now sending the data across. It's telling me I've forgotten a codon. We can engineer around that. The data's been sent into Luma. We're registering each of the individual regions or ingredients, the inserts, the backbones, and then we're registering the association between them to produce the correct construct. And that's all happening in real time. It's always a two-way street though, and I really like actually that I get info directly back here. I get some Luma details here. And as we go through the various registrations, I will get these IDs updated. Okay, so how do I actually go and action this now? How do I use these designs to go and start making these antibodies, start assembling these constructs? And this is where we work in adaptive workflows. So adaptive workflows, the main currency in adaptive workflows are tasks. And these tasks are important for a number of reasons. And you can see that these tasks that I've got configured here follow roughly my antibody journey. I have expressions, purifications, etc. These tasks understand the scientific context. They understand the data I just sent from Geneious. They understand how to manipulate that data and to produce an output. The output of one task can be the input of another, and that can either be very prescriptive or it can be a bit loose if you have multiple ways to get to the same endpoint. And perhaps the most important is these are designed with low code. So you don't need to be a computer science major. You don't need to write SQL. You don't need lengthy consultancy projects to keep pace with the science that you're doing. You just need a savvy scientist that can learn this system. And I'll show you this low code configuration in the next step. Let's go and show you the runtime though. Just the first couple of steps in here. I'm not going to show you the whole workflow. The first thing is I need to order a plate of the inserts, the first ingredient, if you will, from my design. In all of these tasks, we have a few common tabs here. The task tab is really the metadata tab. Gets me to do things like assign, set the status. The operation tab is now where I take the input from my previous step. So I need to find the designs which I've just sent. So if I scroll down to the bottom here, here are the designs that I just sent from Geneious on today's date. Now what it's doing is it's finding that set of designs, it's finding all the discrete constructs, and then it's looking through for all of the unique inserts that were synthesized in that tool. And it's going to allow me to fill them over this 96 well plate. So it's found 106. I'm going to target 10 microliters per well. And the direction that I fill them in isn't really important to me right now. It's very important in the next step that I match this up with the correct backbone. So I'm going to drop these in. So now I have my unique inserts over my plates with the naming scheme that came from Geneious, all linked back to those designs in Geneious. Now, you guys may do this with a CRO, you may do this in-house. You can use the export button here to produce a flat file to send to a CRO, for example, a manifest of what you want them to make. Or in future versions, you'll be able to use this to invoke an API call for some automation, I guess, both internally or externally. We'll imagine some time has passed and I've got my plate in hand, but I need to check, did I get the right volume? Did I get my 10 microliters per well? And to do this, I can import data from LabConnect. Now, I'll show you LabConnect later, but just know that LabConnect sits on top of instruments that produce files or software that produces files. Those instruments could be screening detectors. They could be anything really. And in this case, what I'm going to do is zap in the barcode of each of the plates which I've just received. It's going to go and interrogate all of the data in the system, and it's going to say, hey, there's liquid handler data for these barcodes. Do you want to decorate these plates with that liquid handler data? Once that's done, I've got now my actual volume in here, and I can go back to my operation tab. And actually, if I sort my table here by my actual volume, I get a heat map. And this heat map tells me when there's discrepancies between what I planned to make and what I actually made or what I actually ordered. So at this point, I have my information in hand. I can go and register the batches of inserts. This is now interacting directly with the registration system to generate IDs for me. Let's go and complete this task, since I'm going to use this plate as the start point for the following task, which is going to be adding in the backbones to create the constructs. So I'll go through this one rather quickly. I'm going to search for my clone constructs task. Same deal as before, metadata. Operation is then me using my previous output. In this case, though, all I'm looking for is the plates rather than the design. So I'm pulling in the plates of inserts which I just generated. And now my job is to fill it with the correct vector backbones, to match them up according to that design in Geneious. Notice, though, there's no fill direction here. I don't need to worry about that. It knows which ones go together. It understands that Geneious data, and I can fill that up, make my plates. I can, of course, import data into here. I can register these, and I can move on and basically carry on down my workflow. Now, we're skipping quite a few steps here, I realize this, but let's move on to the next part, which is where we have to take these constructs that we just made and we have to co-express them to give us our crude antibody that then gets purified. And rather than show you the run mode for adaptive workflows, let me show you the configuration. Let me show you that low code input. So I'll go to my task templates. I'll leave without saving. And let's go to my co-expression of my 3 constructs. So again, this is quite complicated, right? Because I have to marry up the constructs with the right protein designs from before. But we do all of this with low code. So my first tab is setting up the metadata. I can choose the statuses. I can choose whether I'm creating a new container, a new plate, a new vial, or I'm using the source of a previous step, as I just did. I can choose whether I can load data from LabConnect. And there's a little teaser to what comes towards the end. I can choose how I want to interact with Prism from these plates that I'm making. The materials tab, I think, is where it gets really interesting, though. I have several materials with several different roles. The first material is really a virtual fill. It's a design role, which basically says, tell me which of the proteins, the full antibodies you designed before, and I will then automatically for you marry up the correct constructs together as inputs to give you your full antibody at the end. For each entity, I can choose kind of how we operate on them, whether I'm transferring them, using them as a target, whether I'm pooling. And my expressing cell line is my output. I can also choose attributes, both displayed, so things from my registration system, metadata I want to help guide me, or recorded, things that I'm adding in, like my actual volumes, my actual amounts. So you can do all of this in a low-code manner and keep pace with the science in your lab really, really simply. Now, before I move to the next stage, everything is tracked in here. As Kaleem said, all of this is tracked by nature of using our adaptive workflows, our design tools, and our registration systems in harmony. I can go and track everything that's happening. So let me go to a different app where I'm doing some tracking. Let me go to one of these experiences and go to set exploration. So what I can do here is I can say, starting at my protein design set, here's the protein design set, which I started maybe last week. Go and show me where I've got to in my wet lab workflow. Show me what entities I've produced at the concept level. Show me those entities that have batches. And then allow me to drill down into the chain level, for example. Go and look at the sequences live in this Geneious sequence viewer that we've embedded inside this experience. But perhaps more importantly, tell me where this came from. What other entities were invoked in this? What's the lineage? And this is all happening automatically. The user is not having to make any hard linkages here. This is being built up by nature of walking through these workflows. The green entities are the concepts, and the blue entities are batches thereof. And we'll be continuing to expand this plugin as we get more and more feedback from customers. But all of this is happening automatically, and I think that's a really powerful message to understand. Okay, so now we're into the test part of our life cycle. So we've purified our antibodies. Now we need to characterize them, and we need to see how effective they are as drugs. And this is where the kind of variety of data starts to explode even further. We could get protein metrics characterization data. We could do screening data in Prism. We could be using flow cytometry in Omic and FCS Express. It's where we really start to have this great data flow that I talked about at the end, at the start, to enable the kind of workflow that I'm showing you today. The first tool that I wanted to talk about was Prism, but let me first just explain that important relationship between the apps in Luma. So the first antibody campaign dashboard that I showed you is on the right here. Each of these white boxes is an app in Luma. I want to draw your attention though to these left-hand section. I call these foundational apps, and as you'll see, they map almost one-to-one with a scientific app, an instrument, a third-party system. The job of these foundational apps is to take data from Prism, for example, to parse that data and put it into a data model that makes sense so that I can use that data elsewhere. We then have these green arrows, and they should really be bidirectional, but these are app-to-app workflows. These allow us to take Prism data and put it into a campaign dashboard. They allow us to take Geneious data and flow it into a materials app where it gets registered and then flow that up into our dashboard. So let's talk about how we get data from an app into one of these foundational apps so that we can invoke it in our workflow, as I've showed you earlier. And let's start by talking about Prism. So Prism is an amazing tool. It's beloved, it's great, but there are a few challenges, or really two main challenges. The first one is getting data in. I call it the arts and crafts project, where you're copying from your instrument file, you're pasting it into Excel, you're pivoting it, you're moving it around, you're calculating, and then you paste it into Prism. Once you're in Prism, you do your workflow, and then you're copying and pasting again to get it into a database or a notebook, or to copy the image out into a PowerPoint. So we're removing that. We're making Prism, along with a lot of these other apps, more enterprise ready, but we're not removing the secret sauce that people love about these apps. And I think that's a really important approach. The reason I have Lab Connect highlighted here as well is that Prism deals in files, Prism produces files, and Lab Connect is where we can handle these files. So let's take a look at how we can break down those two challenges around Prism. First one is, how can I get data into Prism? Now, remember, in adaptive workflows, I can generate plates. Here I've generated 384 well assay plates. I've already gone through my import step. Apart from this time, I've imported detector data into my plate. And I want to show you this new data processing piece that we're working on. So part of the workflow into this percentage inhibition, this IC50 assay, whatever it is, might be to normalize the data. And I can do that by adding a column in here, giving it a name, and then I can type in my expression. So we have access to all of these maths functions, as well as, with the little pounds in front of them, all of the data that Adaptive Workflows is tracking about these plates and wells. I can add filters in here, so I can only do this calculation over a certain well type or group of wells. And I can even group by certain things. So group by the plate, group by material, group by the batch, however it is. And then these will then get incorporated into a template that you can reuse time and time again. So I've prepared the data that I want to put into Prism, and then I can export it just by simply saying, well, what is my X? What is my Y? And how do I want to group this data together? And export. And there, that little icon there, that's the Prism file being written. So I now have a Prism file with all of this data, no copy and paste. That's great. So once we're in Prism, what can we do next? So this is Prism. I've sent some data across into my XY table. It's automatically filled out my info tab for me to tell me where this came from. I've done my curve fitting. I have my curve. How do I get the data back out again? And much like you saw with Geneious, we have logins to Luma. This time we log into a system called LabConnect, since Prism deals with files and LabConnect handles files. So let's have a look. After I press this button, what happens in the journey of this data? How does LabConnect help us in this journey? So let's go out and have a look at LabConnect. So for those of you who've not seen LabConnect before, this is the web-based GUI. Each of these rows you can think of as either an instrument that has our agent connected to it. So we're sweeping up these files constantly, putting them into the cloud here. Or it's a virtual folder where a human can drop files, or a piece of software just like Prism can drop files. So we make this more fair data compliant. We make it more findable and accessible just by nature of centralizing it, making it available through the GUI or via the API. But how do we make it more interoperable? And to do that, we actually parse these files. As Colleen mentioned before, we have parsers for simple files like Excel, CSV, text files, and also for proprietary files like our Prism file, like many instrument vendor files, your mass specs, your LCMSs, etc. So let's go and have a look at the parsed version of these files. So we have a couple of parsers that run by default. One of them is hot folder, as we call it. This gives us access to where the file came from and an immutable link to the file. And then we have the specific scientific parsers, as I call them, to get the actual data out of these files. Now, for the computer scientists in the room, this JSON format will be quite familiar. For end user scientists, this JSON format is not that useful. I don't want, as an end user scientist, to have to drill down through this structure to get at my data. And this is where Luma then comes in, because Luma operates on these JSON representations of these files to give us our foundational app. So let's have a look at the foundational app for Prism. So we're back in data. This is, again, in my opinion, the most important thing that Luma does. And let's have a look at some of these configurations that I talked about at the start. So I'm visualizing the data model here in the middle. Each of these blue squares is a table. I have a table for my hot folder information. Remember, this is where the file came from and a download link for it. I have a table for the data, the results that were generated in Prism. I have a table of metadata that comes directly from Prism itself. And I have that info tab that was generated in Prism for me. And finally, I have my graphs. One great thing about this system is we regenerate the graphs as the file lands in our system, so you don't have to copy and paste those either. So I've got this nice data model, but how do I break apart those JSON representations of my files and get it into this data model? And we do that with what we call raw data flows. Raw data flows allow us to operate on the JSON structure that we've produced in LabConnect. And I can write SQL, or soon to be Python, and basically break apart that data and then map it to one of these tables. And this now happens automatically. So I export data from adaptive workflows to produce my Prism file. I do my analysis in Prism, send the data into LabConnect. LabConnect parses that data, tells Luma it has new data that's being parsed. These raw data flows run automatically. The data gets pushed up into the data model. And then that allows us to reuse that data in experiences directly in Luma. So here's that same file that I just showed you in Prism. In Luma, I have my info tab, I have my link out to the file, I have the data, and I have the graph. And then I can use those app-to-app workflows to take this data and associate it with the entities, associate it with adaptive workflows, put it into decision-making dashboards to help me move my project forward, all without copy and paste. So we're making these apps more enterprise ready without having to rewrite them as a web app. Keep Prism the way you love it, but mobilize the data seamlessly. Okay, the next data journey I wanted to talk about is around flow cytometry. And actually, both of these apps, the data flows in kind of a similar way. They produce files, they go to Lab Connect, they get parsed, it gets put into their foundational apps, and then we can flow them forwards. But this is a really interesting case where OMIC and FCS Express both produce flow cytometry data, but they do it in different ways. Luma can also harmonize and normalize this data, and we can do it for flow cytometry, but we can also do it for instrument data. Imagine how powerful it would be to be able to combine data from multiple mass specs, from multiple vendors into a single app and just search for barcode in one place. And regardless of where you generated that data, finding it in one place, use it in adaptive workflows in a consistent way, use it in dashboards in a consistent way. And that's this part of the demonstration. I want to show you how we can harmonize these data together. Let me show you how we generated some of this data. So OMIC is web-based and FCS Express is a desktop tool. So the workflow here from OMIC actually could start life in the Dotmatics notebook or in another notebook that you can connect via API. In this notebook, I was generating metadata in my notebook and then actually using the buttons here, sending this metadata and creating a data set directly in OMIC. All of the data below here is then delivered back from OMIC via these workflows that I've talked about. So my data in OMIC gets sent, it sets up, it associates this with my experiment. OMIC actually has a really nice integration with Lab Connect. I've connected a cytometer to Lab Connect, so I'm pulling up my raw files. OMIC can just read them directly in one place. Then we build our workflow in OMIC. This workflow moves through auto-gating. So auto-gating allows you to basically speed up and make more consistent this kind of art of drawing these polygons or these gates around this data. And then at the end of my analysis, just like I showed you in Geneious, just like I showed you in Prism, I can then sync this data over to Luma, to Lab Connect, parse the file, push it to a foundational app. FCS Express is a desktop tool and it can do similar things. It can also export data to Prism, just like OMIC. It can also export data to Luma, just like OMIC. And I've actually analyzed similar data in both of the systems here, and I've analyzed kind of a plate-based approach here. So what I want to show you is not the foundational apps. I want to talk to you and show you how I've built this unified app of normalized data across these disparate systems. So let's go back into Luma. Let's go to our flow apps. So I have my foundational FCS Express app. I have my foundational OMIC app, and then I have this higher order app over the top called Flow Luma, for want of a better phrase. Now in this experience, I can take data from either system and view it and interrogate it in a really consistent way. So this data came from OMIC. I've pivoted out the metadata that OMIC sent to us. And then I've recreated some of the plate views that we saw. So I can look at my well results here. I've actually got a categorical filter here, so I can trellis by that filter. I can look at the Prism charts that I created as a secondary analysis to this particular data. So this is using that Prism foundational app and flowing data up into here. But I can also link out to other data in the system. I'm using a small molecule drug here, so I can drill down into some chemistry data directly from this dashboard. So we've talked a lot about biology. We do support chemistry. Here's a look at a small dashboard. We'd be happy to talk to you about that. That's great. If I go and look now at a FCS Express centric data set, I can use these same objects. In this case, I can go and look at all of the files that I used to create this analysis, including a link back to the original raw data files. I mentioned design, make, test, decide before. You can actually do this in flow cytometry. You can design the panels that you're using in something like EasyPanel, and I can link all of that data in here as well. But now I can interact with the objects in a very similar way. I have plate-based data. I've got hits called in here. In this case, though, I'm referencing one of those multi-specific antibody IDs, the things that I generated right at the start of this demo, and I can drill down into that data. So Luma is the great aggregator. It's also the great normalizer and harmonizer of disparate data for similar techniques and analyses. Okay, we're right back to the end. I won't spend too long on here. We're right back to decide. We're right back to that initial dashboard. We're using our app-to-app workflows to push data in there. We've focused mainly on antibodies and biologics today, but again, we do support chemistry in here. Let's just jump back to that first dashboard, and I'll show you a couple more features in there before we close out. So I'm going back to my, oops, it would help if I could spell, my multi-specific campaign. Let's go back to the experience. And let's look at how we're now collecting data across this entire workflow in a really seamless manner. I have SAR data all the way from the formats that I designed to wet lab data from adaptive workflows, to characterization data from things like protein metric, from flow cytometry, to screening data in Prism. I have the Prism file that I showed you before in here as well. I can chart any of this data. I can pull in instrument data here, other analyses, in this case, size exclusion chromatography. And I can even take bits of functionality from our linked applications, in this case, the Geneious sequence viewer. This is fully interactive, so I can zoom in, zoom out. If this is a plasmid, I can view it in the circular view. And of course, I can pull in flow cytometry results, as I showed you before. So this is where Kaleem ended. I'll end on the same point. Hopefully what you're seeing is this is the most advanced scientific platform ever delivered to the market. We're really only just getting started, though. Come talk to us. We'd love to hear your ideas. Thank you very much for the attention. And if you're interested in learning more, there's an email address on here that you can contact us. Thank you.
Thanks, Phil. And for any questions that were not answered in the chat today, we will be following up with you directly post-meeting. Thanks for everyone that's joined today. This has been recorded and will be transmitted and sent out via email for those that attended and for those that registered. Thanks again. Have a great day.
Thanks.